Good morning, children. This is Mrs. Anjum Aleem from Springfields School, Masabtank, biology teacher for class 10. Welcome back to the revision session. Today we shall start with the new lesson, reproduction. I request you all to watch the video attentively, pay attention, listen carefully and understand the concept. Don't miss any part of the video. All topics are important, so please pay attention. We will first focus on the long answer questions, then in the next video you will learn about the short answers. First question is, how are sperms modified to perform their function? It is a microscopic motile structure composed of a head, neck, a middle piece and a tail. The middle piece contains numerous mitochondria which produce energy for sperm motility needed for fertilization. Tail helps the sperm cell to swim to reach the egg cell. Understand? These are the parts of the sperm. You can see in this slide, head, which is having a nucleus, and a pointed acrosome is present, cap-like structure on the head, which helps in the penetration into the ovum. To enter into the ovum, the middle piece has the mitochondria which provides the energy, and tail helps in the propelling. What are the major functions of the male reproductive system? In males, the reproductive cells are called sperm. The function of the male reproductive system are to produce the sex hormones, to produce and store sperms, and to deliver sperm to the female reproductive system. A sperm cell may join with the egg in a process called the fertilization. Now let us have a quick look on the male reproductive system. As you can see here, the male reproductive system consists of the following parts. A pair of testes, two sac-like structures are present. They are actually protected inside the scrotum and they are present outside the abdominal cavity. Vasa efferentia. Vasa efferentia actually arises from these testes and combine to form the epididymis. A pair of epididymis. Epididymis leads into these long tubes which are here written as ductus deferens. It is called the vasa differentia. Two long tubes. These are called as the vasa differentia. A pair of seminal vesicles. You can see here seminal vesicles are present. Can you see here? Seminal vesicles are present. Ejaculatory duct. Ejaculatory duct, can you see? Yes, prostate glands. And of course, Copper's gland is present. These are the parts of the male reproductive system. So, testis produces the sperms which are stored in the epididymis till the maturation. And they are nourished by the sperms are nourished by the testis. They are stored in epididymis till they are matured, then they are released at the time of ejaculation. Now let us see. Testis produces sperms and testosterone. Epididymis matures and stores sperms. Sperm duct, ejaculatory duct, you can say, it carries the sperms from the epididymis to the urethra. Through the urethra they are ejected out. Seminal vesicles, Copper's gland and prostate gland produces seminal fluid which feeds the sperm and allows them to swim. Sperm and seminal fluid are collectively called as a semen. So what is ejected out? That liquid is called as semen, which contains the sperm. Urethra allows the passage of either urine or sperm. Penis places sperm inside the body of a female. Scrotum keeps the testis at a lower temperature. This is the optimal temperature for meiosis to occur. That is the reason why the testes are outside the abdominal cavity in the scrotum. Now let us learn about the female reproductive system. The functions of the female reproductive system is to produce the female egg cells, transport the egg to the site of fertilization. The fertilization of an egg by a sperm occurs in the fallopian tube. I will tell you what it is. After fertilization provides a place for the baby to develop, that is your uterus. If fertilization does not take place, the system is designed to menstruate. Produces female sex hormones and that maintain the reproductive cycle. Finally, the female reproductive system consists of a pair of ovaries, along with a pair of oviducts, uterus, cervix, vagina. The external genitalia located in the pelvic region. Now let us see here. Can you see here? Yes, we will start with the vagina, which leads into the cervix. There is a pear-shaped structure which is called as the uterus. At the top, can you see the sac-like structure? Pair of ovaries are present. Of course, oviduct or fallopian tubes having the funnels at their end are also present. So these are the parts of the female reproductive system. Now let us learn about the function of each part of the female reproductive system. Ovary to produce the egg, to produce the hormones. What are the female hormones? Estrogen and progesterone. Fallopian tubes catches the egg from the ovary and transports it to the uterus, site of fertilization. Uterus, the site of implantation, means the embryo will come, the zygote will come and get attached to the walls, endometrium, the inner lining of the uterus, and this process is called as the implantation. And the release of ova is called as the ovulation. And the attachment of the embryo to the walls of the uterus, that is endometrium, it is called as the implantation. So uterus holds the developing embryo. It has a lining called the endometrium which enriched with the blood vessels to nourish the embryo. So placenta is formed to hold the baby. It acts like a bed. Vagina allows the entry of sperm and the exit of baby at birth. Now, when we are learning about the embryo, we should also know about the extra embryonic membranes. There are 4 extra embryonic membranes in humans. Number 1 is chorion, surrounds the extra embryonic membranes and provides the gas exchange. Diffusion takes place. Amnion, chorion is the outermost. Amnion provides the protective layer around the amniotic fluid. The fetus is present inside the fluid called as the amniotic fluid. Yolk found below the embryo. This cavity is where blood cells are formed which travels to the embryo. Of course, allantois is a part of the umbilical cord in the form of blood vessels to move nutrients and waste. That means, the baby is attached to the mother by umbilical cord. Allantois is a part of that umbilical cord. When the baby comes out, it is cut and separated from the mother. Now let us learn something about the plant reproduction. How does the natural propagation and the artificial propagation? We will learn here about the artificial propagation. This is very important question. Sometimes they ask you questions based on cutting, grafting, layering, like that. These are the different types of the artificial propagation. A plant cutting is a piece of plant that is used in a horticulture for vegetative propagation. A piece of the stem or root of the surface plant is placed in a suitable medium such as moist soil. Some plant can be grown from leaf pieces also, called the leaf cutting. So what are we doing? We are cutting a part of a plant. Either it can be a stem or root and placing it into a moist soil so that it develops into a new individual. Examples are rose and hibiscus. Then what is layering? Layering is also another type of artificial propagation where the development of roots on a stem while the stem is still attached to the parent plant is called layering. Can you see? One of the stem is getting attached to the soil and thereby it is developing new roots and stems. And again one of the stem from this plant will get attached to the soil and develop the roots. Like that they get spread. Our garden grass, it is by this method it grows. Some plants propagate naturally by layering, but sometimes plant propagators assist the process. Nerium and jasmine. These are the examples of the layering. We have seen the cutting, we have seen the layering. Now let us see what is grafting. Grafting is a technique that vegetatively joins the two plants into one. Yes, children. Whenever we want the desired characters, we go with grafting. We select two plants. One plant is called as the stock plant, which is the parent plant we can call. And another plant we can use of our choice and attach both of them by wrapping them with a sellotape or cloth twined. It is twined around it. Now these two are joined. Stalk is the one which is present in the soil and scion is the one which is brought from another plant and it is joined, the part of the plant. Both of them will grow into one. So let us learn about it. Instead of cross-pollinating two plants and producing a hybrid seed, grafted plants use the root and the bottom portion of one plant, that is called as the stalk, and attached to a tender shoot, scion, from the top portion of another plant. Mango, citrus, apple and rose we are all producing these by the process of grafting. Mango, suppose one stalk, the size of the mango is large. In the scion plant, it is tasty, it is very sweet. So if you join them both, you can get your desired mango, very large size and also sweet. Now let us see another type of propagation, that is the spore formation, that mostly occurs in fungi. For example, rhizopus. Spore formation is a method in asexual reproduction. It generally happens in non-flowering plants such as fungi and bacteria. In this, the parent plant generates thousands of spores which individually grow in a plant. These spores are microscopic and round in shape and grow in suitable condition. Have you observed the bread molds? Yes, if you observe them under the microscope, you will observe them like this. The round ball-like structure which is called sporangium, in which spores are present. And then stolon is there, they are called as hyphae. The stem portion is called as the hyphae. Now there is an experiment based on this. Examine rhizopus or common mold under the microscope. Write your observation. For these children, there are two things. First thing is how to develop the molds on the bread. It is very simple. I have written it in the PDF, these answers. Kindly go through there. You have to take the bread, moist it with drops of water, put it in a plastic bag, keep it in the cupboard for dark. Again take it out after 2 days and sprinkle more water and again store it in the cabinet. Like that, after 1 week you will find that the bread mold has developed. Now cut a piece of the bread mold. So here the aim is observation of rhizopus or common mold under the microscope. Materials required is mold sample, bread, plain glass slide, cover slip, water, disposable gloves, and the procedure is: place a drop of water in the center of the slide. Using a toothpick, scrape very little of the mold and place it on the drop of water. Take the cover slip and set it at an angle to the slide so that one edge of it touches the water drop. Then carefully lower it over the drop so that the cover slip covers the specimen without trapping air bubbles underneath. So you have to be careful. Precaution is air bubbles should not be formed under the cover slip. Use the corner of the tissue paper or blotting paper to blot up any excess of water at the edges of the cover slip. View the slide with a compound microscope. What are your observations? Your observation is the common bread mold consists of fine thread-like projections called the hyphae. Yes, can you see here? Hyphae. And thin stems having knob-like structures called the sporangia. Each sporangia contains hundreds of minute spores. When sporangium bursts, the tiny spores are dispersed in air. Whichever place they fall on, they develop again new hyphae and new sporangium. Like that, the life cycle continues. Now let us see the LS of a flower. Draw the LS of a flower. Common parts of a flower. If they ask you to draw, what should you draw? You know that the male reproductive parts of a flower are stamens, that is anther and filament. The anther will be bilobed and it will contain the pollens in it. So pollen are the male gametes. And the female's reproductive parts of a flower are stigma, style and ovary containing the ovules. There are also colored petals present, and at the base of the flower green leaves are present which are called as sepals. Okay children? Now remember, the male reproductive parts are also called as androecium and female reproductive parts are called as a gynoecium or pistil. Now coming to the structure of ovule. Here the ovule is having a first part, it will have the first, this is actually a female reproductive part, pistil, having the stigma, style and ovary. Inside the ovary, the ovule is present, which is present on the thalamus, the base. Can you see the ovule? Yes. The upper end of the ovule is called as a chalaza and the lower end is called as a micropyle. Now, we will see how the fertilization will take place. The pollen will get attached to the stigma of the female part, and it will develop the pollen tube, and through the pollen tube the pollen will roll down and enter into the female cell or sex cell or female gamete ovule. Pollen is coming through the pollen tube. Can you see the pollen tube in the slide? It has two nuclei. So by the time the pollen with the two nuclei enters into the ovule will already undergo 3 times the mitotic division to form the 8 cells. I will tell you what are these 8 cells inside. Okay? It is like this. The first 3 anterior end cells are called as the antipodal. I told you, the top end is called chalazal end and the lower end is called as the micropyle end, from where the pollen enters. So the first 3 cells are called as the antipodals. The central cell is called as the polar nuclei, and the lower cells 2 are called as synergids. In between the X cell is present. So these are the 8 cells ready for the pollen to enter. When the pollen enters, 2 nuclei are present. One will unite with the X cell shown at the bottom, and the other will unite with the polar nuclei, forming 3N. 2N is already there, one more N. So 3N, it is called as the endosperm. Whereas one pollen will unite with the egg, forming the zygote. So this process is called as a double fertilization because 2 nuclei are fertilizing at the same time. One nuclei of the pollen fertilizes with the polar nuclei, forming the endosperm 3N in the center, and one nuclei of the pollen will unite with the egg cell, forming the zygote. The process is called as a double fertilization. This endosperm will provide the nutrition to the developing zygote. Double fertilization involves 2 sperm cells. One fertilizes the egg cell to form the zygote, while the other fuses with the 2 polar nuclei to form the endosperm, as I told you just now. After fertilization, the fertilized ovule will form the seed, while the ovary will form the fruit. So when you eat an apple, you are eating the ovary. The seeds are the ovules. See again here, there is one slide showing the zygote formation. This is there in the textbook and also in your PDF notes. Kindly go through it. I hope you all understood the concept. I wish you all good luck for your preparations and request you all to complete the learning part in 1 week. One lesson in 1 week, so that you are prepared for your upcoming pre-finals and board exams. Thank you very much for watching this video.